Hello friends, today we would be learning principle of inheritance and variation, which is class 12th chapter from biology. So let's get started with the video. Inheritance is the process by which characters are passed on from parents to progeny. Mendel conducted hybridization experiments on garden pea for 7 years. How many years? 7 years, and proposed the laws of inheritance in living organisms. Who proposed the laws of inheritance? It's Mendel. Gregor Johann Mendel. The first hybrid generation is also called filial progeny or F1. Okay. In a monohybrid cross, the phenotypic and the genotypic ratio is in F2 generation is 3:1 and 1:2:1. Okay. In F2 generation, it's 3:1 and 1:2:1. And what are these? These are phenotypic and genotypic ratios. According to Mendel, characters are controlled by discrete units called factors, and now we call them as genes. What we call factors as genes. Genes which code for a pair of contrasting traits are known as alleles. For a character like height, TT and TT are called genotype of an organism, while the descriptive terms called tall and dwarf are the phenotypes. Like I would be telling you that you are tall or you are dwarf, then this is phenotypic. But when we represent by small t or capital T or capital T small t, then this is the genotype. Okay. While the descriptive terms talls and dwarf are the phenotypes. Both the alleles can be similar in case of homozygotes, that is TT, capital TT or small tt, can be dissimilar as in case of heterozygous, that is capital T small t, for a particular trait, that is height. During the process of meiosis, the segregation of alleles is a random process, and so there is 50% chance of a gamete containing either allele. A gamete is pure for a character, you can say. A gamete is pure for a character. Okay. The symbols, that is this one is for female and this one is for male, are used to denote the female egg and male pollens respectively. This one is for female and this one is for male. For a trait height, the genotypic type capital T small t has the phenotypic character, that is tall. When an organism showing a dominant phenotype is crossed with the recessive parent instead of self-crossing, then the cross is called test cross. What? Like this. Okay. If the result of the test cross shows half of the progeny with dominant trait and the half of the recessive trait, then the unknown genotype of the parent is interpreted to be heterozygous. In incomplete dominance, F1 has a phenotype that did not resemble either of the two parents and is between the two. In incomplete dominance, in between. In incomplete dominance, the phenotypic ratio is 1:2:1 and the genotypic ratio is also 1:2:1. Here you can see, like I am crossing this. So what you get is, here you can see, or if I would, okay. So this was that. A study on plant Antirrhinum sp is a good example of incomplete dominance, and that of ABO blood group in human beings is an example of codominance. ABO blood group codominance and Antirrhinum sp example of incomplete dominance, and the ratio is 1:2:1 in incomplete dominance. And learn the definition. When an organism's the recessive parents instead of self-crossing, then this is called test cross. Okay. In Klinefelter syndrome, there is an additional copy of X chromosome resulting in a karyotype of 47 XXY. In Turner syndrome, there is absence of one of the X chromosome, that is 45 plus X0. The traits that are generally controlled by 3 or more genes are called polygenic traits. 3 or more genes are controlling a certain character or a trait. When a single gene exhibits multiple phenotypic expression, exhibits multiple phenotypic expression, this is called pleiotropic gene. What we called it? Pleiotropic gene. Single gene, multiple phenotypic expressions, called as pleiotropic gene. In honeybee, females are diploid having 32 chromosomes and males are haploids having 16 chromosomes. This is called haplodiploid sex determination system. Color blindness is sex-linked recessive disorder. This defect is due to mutation in certain genes present on X chromosome. Thalassemia is autosomal recessive disease. It is controlled by two closely linked genes called HBA1 and HBA2 on chromosome number 16 on each chromosome 16 on each parent. Autosomal recessive disease thalassemia, and the genes are HBA1 and HBA2. Color blindness is sex-linked recessive disorders due to mutation in the genes present on X chromosome. And in honeybee, what we see is haplodiploidy. Females are diploid and males are haploid. In case of codominance, F1 generation resembles both parents. The phenotypic ratio of Mendel's dihybrid cross is 9:3:3:1. What? 9:3:3:1. In 1900, three scientists, De Vries, Correns, and von Tesmack, independently discovered Mendel's result on inheritance of character. Sutton united the knowledge of the chromosomal segregation with Mendelian principle and called it as chromosomal theory of inheritance. Who called it chromosomal theory of inheritance? It's Sutton. Okay. Morgan coined the term linkage to describe physical association of genes on chromosome and the term recombination to describe generation of non-parental gene combination. Morgan used the term linkage, physical association of genes on a chromosome, and the recombination to describe generation of non-parental gene combination. And he used Drosophila melanogaster, that is the fruit fly, for his experiment. The chromosome involved in the determination of sex are designated as sex chromosome, and the rest of the chromosome are called autosomes. Grasshopper is an example of X0 type of sex determination in which males have only X chromosome besides autosomes, whereas females have a pair of X chromosomes. Female are having a pair of X chromosome and males are having only one X chromosome in grasshopper. In mammals, including man, XY type of sex determination is seen. What type of? XY type of. It's seen where both male and female have same number of chromosomes. In mammals, including man. X0 and XY type of sex determination mechanism shows male heterogamity. X0 and XY type. So male heterogamety, whereas ZW sex determination in birds is designated to be an example of female heterogamety. Female heterogamety is seen in birds. In birds, the female have ZW sex chromosome, whereas males have ZZX chromosome besides autosomes. The mutation arising due to change in a single base pair of DNA is known as point mutation. What it's called? It's called point mutation when a single gene is affected. The chemical and the physical factors that induce mutations are referred to as mutagens. What they are called? They are called mutagens, the chemical and the physical factors. An analysis of trait in several generations of family is called pedigree analysis. In pedigree analysis, these three symbols are used. Here you can see affect individuals, and the symbol, this one represents consanguineous mating between the relatives. Sickle cell anemia and phenylketonuria are the examples of autosomal recessive traits. Sickle cell anemia and phenylketonuria, autosomal recessive traits. Myotonic dystrophy is an example of autosomal dominant trait. Myotonic dystrophy, autosomal dominant trait. Example, hemophilia is sex-linked recessive disease in which heterozygous female may transmit the disease to his sons. Sickle cell anemia is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin. In phenylketonuria, the individual lacks an enzyme that converts the amino acid phenylalanine into the tyrosine. Failure of segregation of chromatids during cell division cycle results in gain or loss of function called aneuploidy. In polyploidy, there is increase in the whole set of chromosome in an organism. Down syndrome is a genetic disorder due to presence of an additional copy of chromosome number 21. What we see in Down syndrome is the trisomy of 21st chromosome. So this is all about this video. I hope you like the session. Do like, subscribe to my channel. Thank you.